Hello and good morning, everyone. My name is Miguel Dominguez. I'm with Mirus Bio. I'm the Global Distribution Manager, and I'm joined by Alicia Coop, our Technical Support Manager. And today we're going to present to you transfections that don't suck, ways to increase your efficiency and reduce toxicity. So let's get started. First, I'd like to start off with a short introduction and history of Mirus Bio. We do have over 20 years of nucleic acid delivery expertise. We first started this journey through our Transit LT1 transfection reagent. Over 20 years ago, we launched this product. It was the first serum-compatible, low-toxicity delivery reagent for plasmid DNA. Still continues to be one of our best-selling, one of our most popular products in cases where you're really cognizant about cell viability and toxicity. Transit LT1 is a great option. Through the years, we've developed many other transfection solutions, but more recent, our blockbuster reagent is the Transit X2 dynamic delivery system. This novel polymer-based reagent can deliver not only just DNA, but also small RNAs such as siRNA and microRNA for knockdown studies via RNAi, and it can also deliver CRISPR-Cas9 components, including the Cas9 RNP complex, for those of you doing CRISPR experiments. In addition to that, we also have support for electroporation, or the use of electrical current to deliver nucleic acid. Many of you indicated in your pre-registration questions that you also use this type of delivery method for your transfections. So please keep in mind our Ingenio electroporation solution and cuvettes. We offer these as standalone solutions or as part of a kit. Electroporation solution itself is a broad spectrum solution in the sense that it can transfect or electroporate many different cell types. In addition to that, the cuvettes and the solution are compatible in many conventional electroporation machines available on the market, including the Lonza Maxa Nucleofector 2, the Bio-Rad Gene Pulsar, and the Harvard VTX. However, coming new this year will be our own instrument called the EasyPrater. With this instrument, it will be compatible with our Ingenio electroporation solution and cuvettes. Now we'll be able to offer a fuller electroporation workflow for all of those of you performing electroporations. In addition to that, we'll have optimized electroporation protocols for various cell types. In addition to that, many of you also indicated virus transduction as a platform that you readily use in the lab. For that, we have our Transit Lenti transfection reagent that can generate high-titer functional lentivirus in both adherent and suspension cells. In addition to this reagent, we recently developed our Transit Virus Gen transfection reagent. This reagent not only generates high-titer functional lentivirus, but also AAV in both adherent and suspension HEC293 cell culture formats. So with these different types of delivery methods in place, including chemical transfection, electroporation, and enabling scientists to perform virus transductions, we can now help and enable scientists to program the genome. We can allow you to add, delete, or modify any gene at will. We're also committed to any of the delivery methods to deliver any nucleic acid into any cell type. Now, with that brief introduction, I'd like to hand it over to our Technical Support Manager, Alicia Coop, for our first polling question.
All right, thanks everyone for attending today. Our first question is this: what brought you to this webinar? Please select one of the following. I can't get my transfections to work. My transfections work, but I want better results. I haven't done transfections in a while, so I need a primer. Or what's transfection again? I'll give you guys a second to vote. All right, I think everyone's had a chance. We're almost there to vote. All right, we'll close this and look at what our results are. Okay, thank you for your answers. It looks like most of you are here because you want better results, which is perfect. Miguel will go through techniques to help with exactly that throughout the webinar. For those of you who aren't sure what transfection is, he'll explain that in the very next slide. Just a reminder though, if you would like to ask us any questions, please do so at any point during the webinar through the control panel on the right side of your screen. If you don't see a control panel, look for an orange arrow. If you click that arrow, it'll expand the panel, and then there's a question section, so you can type us questions at any point. All right, back to you, Miguel.
Thank you, Alicia, and thanks to all of you for responding to the poll. Now let's get this webinar going. So what is transfection? For those of you that really want to know what transfection is all about, by definition, it is a non-viral delivery of nucleic acids that include DNA or RNA to mammalian tissue culture cells, although we can also support insect cell transfection as well via chemical or mechanical methods. So just to kind of more specifically tear apart the definition itself to really get us thinking about this process and about this methodology, it is non-viral delivery of nucleic acid. Viruses are very efficient means for delivering nucleic acid into cells. However, it's much longer workflow in actually making the virus, titering the virus, harvesting the virus itself. And in some cases, it does present a higher biosafety level concern. With transfection, we can actually mediate some of these concerns. The biosafety level is not as apparent. In addition to that, it's more turnkey. You can essentially complex your nucleic acid with the transfection reagent, add it to cells, and just walk away. One thing to keep in mind is that many people primarily use it for plasmid DNA delivery, but we also want to call out the fact that many researchers also use it for delivering sRNAs or microRNAs for knocking down gene function. In some cases, you don't need to necessarily knock out a gene to show its function. You can just knock it down transiently through RNAi as well. Alternatively, if you're looking for gene expression, in particularly hard to transfect cells, sometimes messenger RNA poses a different strategy. In regards to mRNA, we only need delivery to the cytoplasm as opposed to the nucleus for gene expression. Many of our researchers have employed this strategy when dealing with hard to transfect cells. And transfection can also be used on a broad spectrum or many different types of mammalian tissue culture cells. So again, just thinking about the definition, transfection applies to non-bacterial cells. When dealing with bacteria, that term is called transformation. And then keep in mind that it's not just reagent-based. Many people are very used to or accustomed to using a reagent for transfection, but a mechanical method can also be employed. One that many people oftentimes use is electroporation, using an electrical current to essentially electropermeabilize the cell membrane to allow different payload in and out of the cell. This can oftentimes work well in cases where cells are refractory to chemical transfection. And now to bring it full circle, what about virus production? So many of you out there are also employing virus transduction as a delivery method. Keep in mind that in order to generate high-functioning virus titers, you do have to employ transfection into your packaging cell line to deliver your virus packaging plasmids. So then with that definition, let's kind of briefly look at the actual process itself, outline some of the barriers and the reasons why there's a lot of science that's gone on behind the process of transfection itself. So we can think about the different nucleic acid species that we're looking to deliver, such as small RNAs, plasmid DNA, or large RNAs, such as messenger RNA. These can all be thought of as different chemical species or compounds with their own inherent properties. One thing about the transfection reagent is that it must effectively condense the nucleic acid and form a stable complex, or what we call a transfection complex. This typically occurs during the transfection complex formation step, and you're adding these two together and incubating for 15 to 30 minutes. What results is a positively charged transfection complex. It's important to have this net positive charge. The net positive charge then allows localization to the negatively charged cell membrane through an electrostatic interaction. It's at this point that the cell then does some of the work and actually endocytoses this complex into an endosome, which is then delivered within the cell. What next has to happen is endosomal release. Obviously, if the complex is retained in the endosome, it is going to be useless. So with some of the formulations that we've developed at Mirus Bio, we can actually allow for endosomal disruption for proper endosomal release into the cytoplasm. It's at this point that sRNA and microRNA, or even messenger RNA, delivery here is sufficient. However, for plasmid DNA, we have to have nuclear translocation for proper transcription and resulting expression. This last step is less understood. However, we do have experiments ongoing at Mirus Bio to better understand and promote this step. However, we do know that there's some protocol optimization that you can do or considerations that you can keep in mind to effectively increase your transfection efficiency. Within the protocol itself, this includes seeding the cells 1 day prior to transfection, and if at all possible, growing the cells in the presence of serum. This will promote active and healthy cell division, which will then allow for nuclear envelope breakdown and better nuclear translocation of the plasmid DNA. So what makes up these transfection reagents and why are they so important? So historically, and among some of the more popular reagents used are liposomal formulations that employ a cationic lipid and a helper lipid to form multi-layered liposomes with nucleic acid. This formulation is one that's behind the lipofectamine brand of reagents. Although it can be very effective for condensation and delivery, the liposomal reagents can be very toxic to the cells. They can destroy the cellular membrane and organelles within the cells themselves. Many of you noted lipofectamine as a reagent that you commonly use in the lab. I would really think about the toxicity that you might be seeing, and in some later slides, I'll address the toxicity and what might be happening to your cells. In addition to that, or alternatively, people can employ the use of a polymer. A very common polymer is PEI. It's also behind the formulation of PI Max, which I noted some of you have also used in the lab. This can also effectively condense in the liver. However, it does have limitations into the number of cell types that it can transfect. So it may not work well in some of the harder to transfect cell types. However, at Mirus Bio, we actually take a synergistic approach to our transfection reagents. And we employ the use of both cationic lipids and polymers to form lipopolyplexes. These are still very effective in condensation and delivery. And because they're non-liposomal, they're much less toxic. And they still work for multiple cell types. In other words, there are broad spectrum solutions for delivering nucleic acid to many cell types. So with this in mind, one thing to keep in mind is the transfection complex that results after complex formation with the reagent. Some of the characteristics that are important are the size. Complexes are typically anywhere between 40 nanometers to 1 micron. One thing to keep in mind here is that during the transfection complex formation step, this is not a static process. It is a dynamic process where the complexes themselves will be growing and aggregating over time. The complexes become too large, they can no longer be endocytosed by the cell. So it is important to keep in mind that transfection complex formation step. In addition to that, the charge of the complex within the complex is very important. So they should have a net resulting positive charge and a positive surface charge density. And ways to modulate this is by looking at the charge ratio. So by the charge ratio, I mean looking at the N to P ratio, or the concentration of nitrogen residues on the reagent, which are positively charged, to the phosphate moieties on the DNA backbone, which are negatively charged. This is something that you can optimize and alter by changing the amount of reagent per microgram of DNA. Now, the reason this is important is that this is something that can lead to either better efficiencies and or expression. So we can illustrate that in a recent experiment we performed on HEC293T17 cells. I noted that many of you are also working with this particular cell type. We transfected these cells with a GFP encoding plasmid and transfected it with our TransitX2 dynamic delivery system. What we're measuring in this first graph is percentage GFP percent efficiency, or the number of cells that are fluorescent green in the entire population. We then modified the amount of reagent per microgram of DNA from 2 up to 5 microliters per microgram of DNA. In comparing this to cells alone, we see that in each particular condition, transfection efficiency is well over 90%. So one thing to keep in mind here is you may actually opt for the lowest amount of reagent, which may actually lengthen the time you have to use the reagent, and may actually also be something to mitigate any toxicity if it is observed. However, when looking at expression, we can now see we can increase the amount of expression. I noted many of you discussed the fact that you were looking to increase your expression. So many cases where you may have identified a strong transfection efficiency, you may still be able to increase that expression. And here we're measuring that through relative fluorescence intensity. So keep in mind, it's not just about the efficiency, but the expression may be something you might want to keep in mind as well. In another experiment to look at these same reasons why the charge ratio is important, we've transfected K562 cells, which is difficult to transfect immune cell line, with a GFP encoding plasmid using either our Transit 2020 transfection reagent or TransitX2 dynamic delivery system. Again, we're looking at both efficiency here on the left graph, expression via relative fluorescence on the right graph. We're going to first look at transfections with Transit 2020. As you can see on the left graph, we can actually increase the efficiency by modulating the ratio between reagent and nucleic acid with Transit 2020. However, one thing we are noting is that expression is tending to go down in the opposite direction. So this would be a good case for you to look to see where you'd want to find that compromise by balancing efficiency and expression. However, in addition to titrating the reagent, equally if not more important is selecting the right reagent. So when looking at transfections with Transit X2, we're now able to go above and beyond this about 15 to 20% transfection efficiency, closer to 40% transfection efficiency. We're looking at Transit X2 and the best ratio. In addition to that, we can also see that expression can also be optimized within this particular cell type. So what we could see here is the highest amount of GFP efficiency and expression can be denoted by the 5:1 ratio with Transit X2. So again, making sure you have the right reagent and the right ratio is key. So why would someone optimize? Transfection has become essentially a turnkey process for many different applications. Among them, obvious ones include gene expression via plasmid DNA delivery, mRNA delivery. Again, modulating those ratios can get you not only better efficiency, but better expression. In addition to that, siRNA or microRNA mediated knockdown, modulating the amount of reagents in your transfection can lead to better knockdown as well. For those of you carrying out genome editing experiments via CRISPR, in particular when looking at Transit X2 delivery of either plasmid DNA or guide RNAs or even the Cas9 RNP, better optimization could lead to better target and higher cleavage efficiencies. And for those of you doing large-scale protein production in suspension CHO or HEC293 cells, you can also optimize or increase the amount of antibody or protein titer produced. Likewise, for those of you generating either lentivirus or AAV, one thing to keep in mind is that sometimes titrating those ratios can not only lead to better transfection efficiency of your packaging cell line, but you also want to look at the functional titers as well. This is something that we've addressed very well through our protocols with the Transit Lenti transfection reagent and the Transit virus stream transfection reagent. In addition to that, many of you might be working on hard to transfect cells. I also observed many of you are working with primary cells. When working with these types of cells, optimizing is key to better expression and also transfection efficiency. And then, of course, one very common application used through many labs is stable cell line generation. For stable cell line generation, you have to have a recombination event occurring. So, of course, increasing the transfection efficiency will lead to greater success in chances of generating your stable cell line. So these are all different applications that are supported via transfection. Keep in mind that we have much application support, including protocols for many of these applications via our website. So one thing to keep in mind, in addition to just transfection efficiency and expression, or in other words, just performance, is toxicity. So something to note in particular with liposomal-based reagents. So we wanted to see what was happening during transfection and how toxicity mediated by transfection might be affecting the cells. We did an experiment where we took HeLa cells that can be particularly sensitive to transfection, transfected them with a plasmid DNA encoding GFP using either our Transit LT1 transfection reagent or our Transit 2020 transfection reagent. Compare that to transfections with lipofectamine 2000 from Thermo Fisher. 8 hours post-transfection, as you can see, the cells still look relatively healthy. However, at 24 hours post-transfection, the cells, in particular with lipofectamine 2000, are incredibly stressed, if not dying. Cells are rounded up and no longer adhere to the plate. This will also translate to your resulting expression. As you can see here, transfections with Transit LT1 and Transit 2020 cells show an abundance of cells that are viable, but also highly expressing GFP. In the case of lipofectamine 2000, we're only seeing a few cells remaining after transfection. In this case, the efficiency may appear to be high, but that's because we've killed the majority of the cells with this particular liposomal-based reagent. So that's talking about and focusing more on visual toxicity. But what could this actually mean for the cells themselves? So we actually took these set of experiments a step farther, and we went ahead and transfected HeLa cells with a non-coding plasmid, thereby trying to eliminate any toxicity or stress to the cell through expression. And what we did is we elicited a gene expression profile using a QRT-based PCR from a human stress response 96 stellar array assay. We looked at fold change of different stress response genes at 8 hours and 24 hours post-transfections of this naked plasmid. And what you can see immediately, even at 8 hours, when the cells still look relatively healthy, there's still a higher number of these genes being upregulated or downregulated with lipofectamine 2000, as opposed to either transit LT1 or transit 2020 combined. And the number of genes just tends to increase even more at 24 hours when we started to notice visual toxicity. So what does this really mean for the cells, in addition to just visual tox and the genes that are being modulated? One thing that we can then map is stress response pathways from these particular genes. We did canonical pathway analysis to identify these different, potentially different stress response pathways that include anything from cell cycle damage, to p53 signaling, and apoptosis, and many others. Keep in mind, this may be some of the stress response pathways that you might be interested in studying, or that may be affecting some of the pathways that you're looking at yourself. So it's just something to keep in mind when performing your transfections. It's not just about the performance, but the effects that it may be having on the cell itself, either through stress and toxicity. We also looked at this in a variety of other cell types to really balance the expression versus toxicity. We looked at HepG2 cells, THP1, NT2, and HUVEC cells, where we compared transfections with transit LT1 and transit 2020 against lipofectamine. Now we're looking at a luciferase-based assay, looking at expression of luciferase in the colored bars, and then measuring toxicity via an LDH assay, or lactate dehydrogenase. As you can see, we're still able to modulate and balance the performance via expression and low toxicity via the LDH assay. If you're interested in learning more about this particular experiment, we do have a handout that you can see and obtain through the sidebar there. The impact of transfection-mediated toxicity, gene expression, and cytotoxicity analysis of transfection reagents. You can also download this via our website. So now for our second polling question, and I'm going to hand it off to Leisha.
All right, our second question. What transfection-supported application do you primarily carry out in your lab? If you don't see your answer below, please take a moment to write it in the question section. So please vote now. All right, a couple more seconds for voting, and we'll close the poll. All right, I'm going to close and then share results. Okay, so it looks like most of you are transfecting for gene expression or knockdown studies, but we've also got a strong representation from the CRISPR and virus crowds. Optimized transfections will improve every application in this list, so we appreciate you taking the time to tune in. Before we move to the final stretch of the presentation, though, I want to take a moment to address a couple questions from the audience. The first one is 6 hours of serum-free starvation necessary for achieving high efficiency? We don't recommend serum starving cells when you're using our reagents, either pre or post-transfection. In fact, we find that removing serum from cells at the time of transfection has a negative impact on efficiency and expression levels. We also see no benefit to serum starving before transfection, and then adding serum back in at the time of transfection. With our reagents, it's best to just leave the cells in complete serum-containing media for the entire process. Another common question that we're seeing, our lab uses lipofectamine. Do you have any general use reagents that work better? So I'm going to preface the answer with reagent performance is always going to be cell type specific, and there's no one magical solution, but our Transit X2 reagent is nearly always a great reagent to evaluate. It delivers plasmid DNA as well as siRNA and microRNA, CRISPR guide RNA, and RNP complexes to cells, and it is often our best in-house reagent for most cell types, both primary and immortalized. Keep in mind, though, that if you're not sure which Mirus reagent to select for your cells, we have this fantastic app on our website. It's called Reagent Agent. That ensures that you'll pick the right one. So you go to Reagent Agent, you type in your cell type and your nucleic acid, and then it will tell you what we would recommend, and often includes experimental conditions that will help you, start out with the right ratio and whatnot. All right, and back to Miguel.
All right, thanks, Leisha. Just a reminder to all of you, keep in mind that if you have any particular questions that you'd like to ask of us, you can also do that in the questions section of your sidebar there. So make sure you pull that out with the orange arrow so that you can see the questions there. All right, so let's continue on. So as we've been talking a lot about the definition of transfection, what's the process that's undergoing, some ways to optimize the transfection efficiency and expression. So now let's get into other parts of the protocol itself that we can think about optimizing. That includes any media additives that you might have within your cell culture. Also thinking about the nucleic acid itself, and with that, the complex formation time. Things to think about within your cells and your cell culture, and then of course your final harvest, and in particular, what kind of outputs are you using to essentially monitor transfection efficiency and expression. So with that, let's start with some of the additives that people tend to add to their media. Chief among them is serum. So serum, as we've talked about before, our reagents are serum compatible in the sense that once you form the transfection complex, we recommend, if possible, adding doing the transfection in complete media with serum. However, during the complex formation step, when you're adding the nucleic acid to the reagent prior to transfection, you need to keep serum away. Serum contains nucleases that can degrade the DNA or RNA that you're delivering. In addition to that, it has other undefined components that may interfere with proper complex formation. In addition to that, in regards to forming the complex itself, make sure to keep antibiotics out of that step. Anything that's highly charged, any polyanions that may be highly positively charged or negatively charged, keep it away from that complex formation step as well. As we talked about, it is an electrostatic interaction between the reagent and the nucleic acid itself. And of course, any surfactants such as floronic, we definitely recommend keeping all of these components away from the complex formation step. However, when transfecting, obviously you can use antibiotics. If you have to have anything that's highly charged or high amounts of surfactant within the cell culture itself, if you're going to use high amounts of any of these particular components during your cell culture, we ask that you wait at least 4 hours before reintroducing this after transfecting. So again, just to be clear, we want to keep all these components out of the complex formation step, but they can be present in the cell culture in relatively normal amounts. In the particular case of serum, as Leisha mentioned earlier, it is very key to keep this within the cell culture if at all possible. For those of you that are doing virus production, we do find that the presence of serum during virus production does help lead to higher functional titers, so it's something to keep in mind. Now, what about nucleic acid quality? Obviously, you want to make sure that the nucleic acid that you're delivering, in particular with plasmid DNA, is of the utmost highest quality, free of any carbohydrates or lipids that may inhibit complex formation step. So make sure you check your concentration prior to any transfection. Make sure you have the right concentration as well. This will always obviously have a role to play when you're trying to optimize that charge ratio, that amount of reagent to nucleic acid. For determining concentrations and looking at purity, making sure that you have an A260-280 ratio of 1.8. If working with siRNA, microRNA, or mRNA, obviously working in RNase-free conditions to avoid degradation. And when using siRNA, make sure to use a buffer that comes with your siRNA. Avoid using water, which can denature the siRNA itself. And we do have recommendations on our website and our protocols for other buffers you can formulate in-house for resuspending siRNA. One key thing to keep in mind as well is endotoxin. So a particular form of endotoxin, such as lipopolysaccharide, can actually inhibit proper transfection efficiency and expression. So making sure that this is away from your plasmid preps, in particular when prepping from a mini prep kit. Many mini prep kits are not rated endotoxin free. However, in the case of mid-D prep kits, they typically are, but in either case, make sure you check. So here we're just showing an example of resulting luciferase expression after transfecting cells with either a commercially purchased plasmid containing very low level amounts of endotoxin, or EU units per milligram of DNA, versus a crude plasmid prep from a mini prep kit containing high amounts of endotoxin. As you can see here, we have expression from the commercially purchased plasmid, but none, if zero, essentially, from the crude plasmid prep. However, if we then take this actual crude plasmid prep and subject it to 3 rounds of our Mirror Clean endotoxin removal kit, we can now reinstate expression from this particularly crude plasmid that's been cleaned. So you can see endotoxin can play a large role in inhibiting your transfections in proper efficiency and expression. What about modifications to nucleic acid? In the case of mRNA, transfection of mRNA, obviously we ask that they recommend that they be 5' capped and 3' polyadenylated. But furthermore, we also recommend modification of the nucleosides, in particular pseudouridine and 5-methylcytosine. We typically employ these for all of our mRNA transfections. The resulting transcripts are more stable and less immunogenic. This type of modification has essentially enabled us to perform repeated transfections over a 2-week period using a modified GFP RNA into BJ fibroblasts. So in this experiment, we did daily transfections over 14 days using either our transit mRNA transfection reagent or other commercially available products. As you can see from this short video, transfections with mRNA using this modified GFP RNA leads to high expressing and high efficiency GFP efficiency. But not only that, the resulting cells after 14 days, as seen in the resulting phase contrast image, shows nice healthy cells with good morphology. So again, some aspects to keep in mind when modifying mRNA. In addition to that, nucleic acid amount can also be titrated or optimized. Many of you discussed that your efficiencies worked well, but you wanted to increase your performance. So in this particular graph, we're looking at transfections of a luciferase expressing plasmid using transit LT1 across COS7, CHOK1, and HEC293 cell types. In this particular experiment, we identified a 3-to-1 ratio, or 3 microliters of reagent per microgram of DNA is the best ratio for these particular cells. We're delivering 0.4 micrograms of DNA. So in addition to finding out that being the best ratio, now we want to go ahead and increase expression. One thing that we can easily manipulate is the amount of nucleic acid that we're delivering. So in this case, going from 0.4 micrograms to 1 microgram. But what's key to, what's really important to note is that we want to maintain that same ratio when increasing the amount of DNA. However, one thing to keep in mind is that as you start increasing the amount of reagent, the amount of nucleic acid, toxicity may start to become an issue. So it's something that you can try to balance. Maybe you can find a middle ground if you see too much toxicity at a higher end of nucleic acid, or perhaps you can still afford to go higher if your cell is still relatively healthy. When looking at RNAi experiments or gene knockdown experiments, we also kind of have the same thing to keep in mind in regards to amount of nucleic acid delivered. In this particular case, we're looking at COS7 cells that are stably transfected and expressing luciferase. We're knocking down or targeting the luciferase gene with an siRNA, delivering that with our transit TKO transfection reagent. What we can immediately see is we're getting good knockdown at about 5 nanomolars of siRNA delivered. However, going much beyond that doesn't seem to afford much more knockdown. Conversely, what we might tend to see is off-target effects and even higher toxicity at the higher amounts of reagent. So maybe sticking at the 5 nanomolar range is the safest and best bet for your particular experiment. So something to keep in mind there as well. Now, what about complex formation time? So again, complex formation time is the time that you're incubating your nucleic acid with your reagent. For most protocols and applications, that usually is a 15 to 30 minute window. We can actually illustrate this in the first graph here on the left. Here we're showing CHOK1 cells transfected with a luciferase encoding plasmid using our transit LT1 transfection reagent. We transfected, and when we made the complexes, we did a time titration to look at different times for complex formation. As you can see, we're reaching the highest amount of expression between the 15 to 30 minute window. One thing to keep in mind here is typically going over 30 minutes is never helpful. As we discussed earlier, complex formation is a dynamic process. The complexes will start to get larger and aggregate, being not conducive to endocytosis. However, in some cases, going lower in complex formation time may show a benefit. So this is something you consider optimizing within your protocol. In regards to shorter incubation steps, this is definitely the case for transfecting mRNA. In these CHOK1 cells, which are capped and polyadenylated for a luciferase expressing transcript, we're using our transit mRNA transfection kit for delivery. Here we're looking at the optimal time of expression is for complexes formed between a 5 to 10 minute window, so much shorter than the 15 to 30 minute window. Actually, anywhere beyond that 10-minute window, expression starts to reduce drastically. So this is something to keep in mind in regards to mRNA, something that we spell out in our protocol, and we typically recommend not going beyond 5 minutes just to make sure to give you enough time between that window. Next thing to consider is your cell culture itself, your cells, the confluency. Obviously, wanting to make sure that you're actively passaging them, that they're actively dividing, growing in the presence of serum. Here we're looking at a panel illustrating NIH3T3 cells at various confluencies, anywhere from 10 to 30 to 50 and 90% confluence efficiency. One thing to note is that transfecting cells at a very low confluency, much lower than 60%, 50% can lead to high toxicity, but also something to modulate within your experiment. Maybe look at the cell culture confluency itself. Here we're looking at luciferase expression after transfecting NIH3 cells at either 50% confluency using Transit 2020 and Transit LT1 at various ratios, with Transit 2020 topping out at about 1 million RLUs, or relative light units, and with Transit LT1 topping out at about 1.6 million. However, when increasing the confluency, we're now increasing from 1 million to 1.5 million with Transit 2020, and now going over 4 million with Transit LT1, and at 90% confluency, we're achieving even higher expression, especially with Transit 2020, now closer to 3 million, and Transit LT1 closer to 6 million RLUs. And again, looking at the optimal titration or charge ratio is also key to finding the highest expression as well. Now, in regards to your harvest times, or when you're going to harvest your cells post-transfection, keeping in mind your particular application. So many of you are doing gene overexpression studies, which can go anywhere between 24 to 72 hours. For gene knockdown, we go out a little bit longer, anywhere from 48 to 72 hours as opposed to 24 hours. For mRNA translation and then resulting expression, that window is much shorter. It's a much faster window, so anywhere from 4 to 24 hours. So make sure if you're looking at expression from an mRNA, in particular transfecting very hard to transfect cells, that you're looking at a much shorter window. And then in regards to protein production, in the case of large-scale protein production from suspension CHO cells or suspension hectoglobin 3 cells, think about, we typically see production going anywhere from 2 days out to 2 weeks or 14 days. So again, think about your application and resulting harvest time. And just another example where we're just kind of illustrating this time frame post-transfection. We've transfected a number of insect cells, SF9, Hi5, and S2, with a luciferase encoding plasmid. And then we're looking at expression post-transfection with our transit insect transfection reagent. So again, looking at higher expression from 24 to 48 to 72 hours. So make sure you have the right harvest time dependent upon your application. And then output. We always recommend a reporter system such as GFP or luciferase when looking or trying to measure your transfection efficiency and expression. In this case, we're looking at GFP. It's among the gold standard that many researchers use. Here we're looking at high GFP expression, but not only that, we're looking at nice cell morphology across a number of cell types, including some primary cells here in the asterisk panels. This is after transfection with our Transit X2 dynamic delivery system. Again, if we're looking at proper efficiency, we're looking at the number of cells that are fluorescing out of the entire population. And for expression, we can measure that through mean fluorescence intensity. In addition to that, luciferase is another popular reporter system that's used. We oftentimes employ that here at MIRS because it's a much more sensitive assay than GFP. Again, here looking at 2 different cell types, A549 cells and MDCK cells, transfecting these with Transit X2, luciferase encoding plasmid. Again, wanting to balance that expression, denoted by the colored bars, and then also looking at toxicity via LDH assays. Lactate dehydrogenase is an enzyme that's secreted into the supernatant that can be measured by a cell-based assay. Again, we're wanting to strike that balance. It's not all just about performance, but we also want to look at the toxicity. What we're able to see here is this Transit X2 performs much better in both categories over lipofectamine 2000 and 3000. In addition to reporter systems such as GFP or luciferase, something that's much simpler to enable as well is labeling of your nucleic acid. One advantage with labeling your nucleic acid is that you can also track it. So if you're not sure whether the problem with expression might be due to your construct, maybe you're just interested in querying whether or not the nucleic acid is actually getting to the nucleus or even just to the cytoplasm itself, you can easily label the nucleic acid, and we recommend using a fluorophore, a process that's non-destructive, but also non-interfering for the particular application. We do offer our Label It technology. It's a very simple technology whereby you can attach a fluorescent label to any nucleic acid. It is a covalent attachment, so it's very stable. This happens through a 1-hour incubation at 37 degrees, and which can be purified through ethanol precipitation or the spin columns that are provided with our Label It kits as well. And with regards to this application and technology, it is non-destructive to your nucleic acid and also can be non-interfering for your particular applications that you're using. So with that, we'll wrap it up to the third and final polling question. Please take a moment to respond. We do have a few more slides, but we'll take care of this nice last polling question with Alicia again.
All right, thanks. What is your greatest cell culture sin? It's okay, you can be honest. You either forget to passage your cells, maybe you transfect when your cells are over or under confluence, maybe you never optimize or read the protocol or use the same protocol for your current reagent as a new reagent, or you neglect to use controls. All right, so I'll give you guys a few seconds to answer. All right, I'm gonna close the poll and we'll look at results. All right, thank you for your answers. None of us are perfect, and that's totally okay, but it's important that we understand the impact of each sin, so to speak, on our results. Looks like some of us, well, some people, maybe not any of you watching, forget to passage cells, which can lead to senescence or even morphology changes, both of which will impact your transfections. And it looks like a very common error is transfecting cells at the wrong density, or perhaps not always transfecting at the same density, experiment over experiment. As Miguel showed, density at the time of transfection will significantly impact the cell's response to transfection, which, if underconfluent, can lead to cell stress and tox. If overconfluent, expression levels will likely be lower. This audience question actually highlights the importance of a few best culture practices. You asked, do you have any tips for improving suspension cell transfections? I work with leukemia cells. Well, suspension cells are easier to work with because you don't have to trypsinize them. They're often much more resistant to transfection. If possible, we recommend splitting them 24 hours prior to transfection to kind of kickstart that cell division. Another helpful trick when working with these cells is to increase transfection complex dose. And Miguel highlighted this a couple slides back. But for example, THP1 cells, it often helps to use a 2x dose of transfection complex, which means doubling both the DNA and reagent delivered to the cell. And that is, of course, after you find that optimal transfection ratio. All right, and we'll hand it back to Miguel.
All right, thanks, Tisha, and thanks everyone for answering and responding. I'm going to wrap it up here with the last stretch of their presentation. And so the last thing we've talked about, defined transfection, the process that everything that's undergoing through the process itself, ways to optimize, and so one thing that we're really stressing is optimization. As I see, some of you did note that you don't to read the protocol or optimize. We highly recommend that when testing, especially evaluating a new reagent, that you definitely optimize with that particular reagent. Don't just simply swap the protocol. All reagents are not created equal. They're all unique formulations, so it's highly important for you to optimize And we actually make that very simple for you. We do offer free samples of all our transit transfection reagents and also our ingenious electroporation solutions. In the case of chemical transfection and our transit transfection reagents, we also supply this DNA optimization protocol within each of our samples. It's also available as a handout on the sidebar there as well for you to download, or you can go to our website as well. With this nice, simple protocol, we walk you through how to plate the cells, whether they're adherent or suspension, and within a 12-well plate culture format. We then actually outline and call out all of the volumes. We've done all of the math to make a variety of master mixes, anywhere from 2:1, 3:1, 4:1, and 5:1 reagent to DNA ratios, denoting the amount of serum-free media to use, the amount of transit reagent, and the amount of DNA. We've also color-coded all of these onto the 12-well plate for you to easily visualize that. And including that, we also provide a number of wells for controls. At the very least, you want to have a cells-only control, but if possible, a reagent-only, a DNA-only, and a media-only control work very well for your particular experiments. One thing to keep in mind, if you can't use a 12-well plate format, you can scale this linearly according to the surface area of the well. So if you wanted to use maybe a 6-well plate or something smaller like a 48-well plate, you can definitely scale that accordingly to the surface area of the wells. And lastly, we do walk you through how to distribute the complexes to cells, making sure that you add them dropwise to different areas of the well, and to gently rock the plate for even distribution of the complexes. And then make sure you harvest at the appropriate time for your particular experiment. And then on the flip side, we also, just as a reminder, have a few other factors to consider, including cell passage number and confluency, things that we talked about today, DNA quality and DNA quantity. And again, thinking about your complex formation time, that 15 to 30-minute incubation of the DNA and reagent. But again, something that you can consider is going to a shorter complex formation time that might be advantageous for certain cell types. And of course, make sure you consider the post-transfection incubation time. One thing I also wanted to add was a few of you noted as a pain point was consistency of your transfections. One thing to denote here is that you really want to consider your cell passage number and cell confluency. When comparing between results, make sure you have similar cell passage number between the cells and similar confluency at the time of transfection between those experiments. Those could actually lead to differences in expression and efficiency. So keep that in mind. So with that, I'd just like to summarize that Mirus Bio does provide over 20 years of nucleic acid delivery experience to help you program the genome. Keeping in mind that transfection is a non-viral delivery methodology that supplies or supports many life science applications, including CRISPR genome editing, large-scale protein production, high-titer virus production. Again, don't forget we have support for all of these applications on our website and protocols to support those as well. Keeping in mind that reagent formulation is key and very important to many of these applications, and in particular, we're looking or finding or addressing the right cell type. As Lisha mentioned, we do have a reagent agent transfection database on our website that you can query to find the right solution for your cell type. You can just input your nucleic acid and your cell type to find the right solution. Of course, consider all the other protocol parameters that we outlined today, and to make sure to strike that balance. It's not just about performance, but you also want to minimize toxicity. Again, visual tox also means underlying stress response from the cell itself. And then, of course, keeping in mind that charge ratio and optimizing that through our DNA transfection protocol. Keep in mind that's available as a handout, but we can also help you optimize in 3 easy steps. You can download that protocol from our website, www.mirusbio.com/webinar. From here, you can also request free samples of our protocols, free samples of our transit transfection reagents, and of course, you'll have the DNA transfection optimization protocol within there as well. You can download it now. Make sure to use it during your evaluation. And for all of those of you attending the webinar that do go ahead and request a free sample, please use the following promo code, Optimize2018, within the comment section of the sample request, and we'll send you this Programming the Genome t-shirt. And finally, for those of you that have any additional questions, if you want to contact and reach out our technical support team, you can do so directly at techsupport@mirasbio.com or by calling us at 844-MIRAS-BIO. That's 844-647-8724. And with that, I'm going to sign off and hand it over to Alicia for any last minute questions.
All right, thanks, Miguel. It looks like we're a bit over time here, so I'll just address one more question, kind of the basic one that wraps into what Miguel just talked about. What's the most important parameter to optimize for the perfect transfection? All right, so there are many critical steps, as this webinar highlighted pretty well. However, it's vital to start with healthy, actively dividing cells and high-quality nucleic acids. If you're working with DNA, endotoxin-free is absolutely a must. Beyond that, I would say finding that optimal reagent to DNA ratio for your cells is key to getting you the best expression and efficiencies. And with that, we will wrap it up. If you have any additional questions for us, feel free to leave questions in the section or just email techsupport@mirasbio.com, and we're happy to discuss any of your cell culture applications. Thanks so much for your time.